All right. Good morning, everyone. It's a pleasure to be here in this wonderful university, very beautiful place. So this is Modes Analysis of Prediction Games by myself, Tom Willkens, and my advisor, Dr. Jordan Pollack, with the demo lab at Brandeis University. So a bit of background. So we're going to be working with co-evolutionary algorithms here. And the difference between that and a traditional evolutionary algorithm is that interactions occur between different populations as opposed to just having some fixed fitness function. We're going to be dealing with organisms with a genotype of some sort of labeled heterogeneous graph as representing some kind of abstract machine. This could be a neural network. In this case, we're going to focus on finite state machines. And also the kinds of interactions that we're interested in here are broadly speaking cooperative and competitive. And when we have these different populations, they can make up an ecosystem with a particular topology. And so we have an example right here where you can see the orchid mantis preys upon certain species of moth, which has a more or less mutualistic relationship with species of orchid plants. And you can see interesting phenomena such as them evolving to share certain phenotypic characteristics. Some other terminology, we have a complexity metric, so we're evolving complexity here. So some function of an individual's genotypic or phenotype. And we're going to focus on genome size, and we're also focused on open-endedness as a broad concept. Specifically, we're going to be looking at complexity growth. So this is the case where the average complexity of organisms in the ecosystem grows reliably over evolutionary time and exceeding any sort of genetic drift. So high-level questions have to be asked about this sort of thing. Can we reliably induce open-ended behavior in artificial systems? There are many different systems shown exhibiting certain aspects of open-endedness. We're focused here on this growth of complexity over time. And we also need to think about how could we put this to practical use in digital systems, artificial systems, and how can we use this to further enhance our understanding of the natural world. And you can see some examples of some ecosystem topologies right here. So you have two population models with the competitive interactions, cooperative interactions, and we're also going to be looking at some 3 species models. So these are a little bit more complex. The domain that we're going to be studying here is the linguistic prediction game. This was introduced by another of Jordan's students, Nicholas Moran, in 2019. And this is a game similar to the iterated prisoner's dilemma. It's a bit simpler even. So it's an iterated game where at each time step, your agent is going to emit a bit, either a 0 or a 1. There's going to be 2 varieties of the game, cooperative and competitive. In a cooperative game, each is basically rewarded for matching or possibly mismatching bits. So there's a symmetric reward between each of the individuals. Whereas in a competitive relationship, the rewards are asymmetric. You begin without information about the identity of the other or the type of game that you're playing. So the sort of game that you're playing needs to be inferred through the information and interactions that you have with your partner. We're looking at organisms defined as finite state machines, deterministic finite state machines, DFSMs. And you can see an example here on the right of a traditional schematic view of some FSMs where we have labeled transition links and labeled states. And so the bit is basically the label of each state, which then prompts the transition of the partner. And you can see a dynamic representation of this game. The yellow indicates the current state. When it's shaded, that indicates a state or link that hasn't been traversed yet. And so starting at the beginning, you can see that trajectory of interaction building over time until a loop occurs when they both return yellow on that first state. In this case, both would get a score of 0.5. So the final reward is that average score over the loop. So we would be just looking at those steps right before that loop occurs. So In Nick's work, he used a simple set of experimental conditions. This is a pretty traditional coevolutionary setup. Small population size, only around 50. Organisms starting from a single state. So we're starting from a minimal state and trying to see what conditions lead complexity to grow over time. There's asexual reproduction with 4 mutation operators. So you can add a state, remove state with equal probability, redirect a link, and then flip the label of a given state bit. So on the whole, there's no bias to grow, but there will be a tendency to grow over time because you're bounded at a size of 1. There's all versus all interactions. So each member of the population plays against all 50 of its opponents in its respective groups. We use fitness proportionate or roulette selection, if you're familiar with that from more traditional evolutionary algorithms. And our complexity metric, which we're using, is a count of the number of states in the genotype following a structural minimization procedure. So we're using Hopcroft's algorithm. And you can see here our results from Nick's original paper. You can see the control group has a gradual growth. Cooperative group, 2 species cooperative, is similar, whereas the competitive group is more of a flat line. We're also going to be looking at the 3 species mixed, which was proposed to have open-ended tendencies, especially in the mutualistic group, which we'll be looking at in more detail in a minute. So there were some hypotheses that Nick proposed in the original work that were difficult in order to verify just due to the nature of the organisms. These are finite state machines. Over the course of an entire evolutionary history, you're dealing with millions of machines. He evolved them for 100,000 generations. And so it was difficult to sort of quantify or verify the hypotheses that he had in mind. So let's take a closer look at the 2 species cooperative group. So this is after 50,000 time steps of evolution and a reproduction that I did of his experiments. If you're in a 2 species cooperative group, you can see that double arrow on the end. You're incentivized to be as predictable as possible. You both rewarded. If I say 0, you say 0, we both get a point. And indeed, if we look at the dynamic visualization there, even though there are many states, the actual interaction is only 2 time steps long. Both emit a 0, both enter a loop. And so that growth around it is actually a result of genetic drift. So this contrasts with the 2 species competitive group, where we hypothesize organisms should behave more unpredictably to avoid being trapped by the other. So we can see here in that interaction trajectory at the bottom, there's a longer and more elaborate interaction. What's interesting also is that the complexity size is tightly bounded below 25. And so the hypothesis that Nick had was that convention chasing or cycling phenomena was responsible for this cap on growth. So it's something inherent with the domain. Now, the 3 species mixed was the most notable entry in the group, where it was hypothesized that we saw open-endedness, some sort of open-ended characteristics of the system, in particular in the mutualist species. So we could think of that as being like the orchid in the prior example given before. But when I was doing reproductions of this to try to verify, I noticed that the episodes were very short, even relative to the much, much smaller machines in the competitive group. So even though this machine on the right, the mutualist, has over 150 states, its interactions were only 6 time steps long. So this led me to hypothesize that the bulk of the growth could in fact be a non-adaptive byproduct of the evolutionary process. So to test this, I decided to use the modes toolbox introduced by Emily Dolson and Anya Vostenar, the measurements of open-ended dynamics of evolving systems, which is a set of tools and techniques to try to pin some simple metrics onto a hypothetically open-ended system. So we use a persistence filter to screen out neutral clades and maladaptive lineages to only focus on the adaptive lineages that persist over time. And then the second main component is an adaptive pruning step in order to prune those existing genotypes into the elements that are most essential for fitness. So we used our own heuristic. So this was a fitness preserving heuristic that used genetic recency. So we start from the most genetically recent and then prune back from there. And then we use the modes metrics to those persistent pruned individuals in order to get a set of information at the end, which gives us a rough sense of the open-ended characteristics of the system. So change is one metric where the idea is we're looking at each of these checkpoints and seeing of those adaptive individuals, how many of them are different from those at the checkpoint before it. Novelty checks to see how many individuals have emerged that have never been seen before. And ecology is just the Shannon entropy, which gives a sense of how spread out those members of that group are. So is there one type of organism that dominates the group, or is there a more diverse ecology? So taking a look, we're just going to focus on the change and novelty metrics first for the two species ecosystems to check the hypothesis that convention chasing or strategy cycling was occurring. And so we can see that there's a slight rise and increase among the control and co-op groups, which I hypothesize is that as they grow, the space of possible novelties tends to increase. But we notice, especially looking at those pink lines, that's when we apply the adaptive pruning to the cooperative group, which dramatically reduces both change and novelty. So of that big organism before, with only 2 time steps of interaction, all of that bloat wasn't necessary for it to perform in its environment. So when we prune it adaptively, it shrinks down to just a couple nodes. And so that's why we have that much lower metrics. Now, in order to test the hypothesis, we check the slope of novelty and change against each other. Sorry, that's a bit in the way. And what we find is that there's a negative slope in the novelty group that's statistically significant, whereas the change metric of the competitive groups is relatively flat. So in the Modes Toolbox paper, this is evidence of cycling behavior, which gives some support for Nick's hypothesis. For the two species complexity, this is the most important result. So the green group is that, two species complexity. We see that the cooperative group shrinks way down, which was what we believed would happen before, and that the competitive group is actually more complex once the adaptive pruning procedure has taken place. Yeah, now this was the most important one. In the 3 species complexity group, what was hypothesized to be the open-ended group, the 3 species mixed, the green line at the top graph, once we apply adaptive pruning, it drops way down to the pink line, so almost flatlined with roughly equal complexity as the competitive group. So this gives evidence of non-adaptive bloat in the organism. And that's supported if we look at the episode lengths. So just to wrap it up, got 1 minute left. We see cycling phenomena are present in various ecosystems, which supports the hypotheses that Nick had before. It does appear, however, that the bulk of mixed ecosystem complexity is non-adaptive. And so there still is a question of what is responsible for this growth that exceeds the control. There's another domain which I introduced at ALIFE last year, the collision game, which did appear to exhibit some open-ended growth that was resilient to adaptive pruning, but the modes toolbox is much more focused scrutiny. And so we want to see whether or not it stands up. We also have a generalization of the game, the continuous prediction game, which needs to be implemented and tested. And also the idea is to analyze the evolutionary trajectories. And so we have a prototype of a dimension reduction technique using graph neural networks in order to map out the trajectories. So this is actually built using those same finite state machines, starting from, you can see the genesis point at the far right. As they grow over time, they're biased to grow synthetically. You can see they sort of flow through this pipe in the space, interweaving around. So I'm going to talk more about this method at the lightning talk at the Emerging Researchers of ALIFE on Wednesday. Yep. So that's all. Thank you so much for your time. Thank you.